දකින්නත් ඇති එයා යනවා. ඒක එළියේ ඉඳලා ගොඩක් පාස් කරලා. දැන් මොකද මේ මොඩින් කියන ඒක නෝනා කිච්චන්නේ ඒක පුතා छोड़ पाते हैं जो इसमें कौन ज्यादा नहीं किया हम तो वाले इन्हें जाना कर पूरा वही से तो
അവനെ ഈ മാർഗീസ് വായിക്കണം അങ്ങോട്ടേക്ക് വന്നത് after a long time. That's it. So how are things going on? Good. At least around 1 month, right? Good. All are not present today, no? In the company. Ano? Samani, usually. Karwali, ඉන්නවද තව කට්ටිය ලයිව් ෂොට් නේ? මේකෙන් මොකක්ද නැහැ ගලවන්න පුළුවන් එක නේ? එහා පැත්ත මේකද?
මේ පැත්තටනේ සෙල්ලම්.
මේ පැත්තෙන් ගහලා බලන්න. මොකක් කරේ එක පැත්තක් හරියයි. ඒවද? නෑ අනිත් පැත්ත ගන්න. යාවෙත් නෑ. ආවා හරි. Right. Okay. Let's start. Well, we are done with biochemistry, and yeah, The marks are good. I will upload by Saturday. Everyone was good. Some were absent for the exam, right? දෙන්නවා ආවෙන්නේ. ඒක තමයි list එකේ ඉන්න අඩු ගන්නවා. දැන් තවලට පස්සේ මීට වඩා ගොඩක් හිටියා කියලා. I was counting the rows also from my mind. මීට වඩා හිටියා කියලා මේක කරලා. මොකද? very plus. ඒකේ ලේසිත් එක්ක. ලීබා ඉන්න ස්ටාර්. සික්සල් අටෙන්ඩන්ස් උනන්දු. එහෙනම්. ඊගාවෙක් කරමු. අපරාධ it was easy. ඔය අන්තිම විදිහට මම අමාරු ඇති කියන විදිහට question sign කරලා ඊට පස්සේ අලුත් ඒවා දැම්මා කියලා. ඊට පස්සේ assignment එකට දීපේ ඔය question sign කරා. ලේසි එකක් ඇද? මේ same batch එකට ඉන්න set එක අනුව තමයි. හරි. So that is that. Now we are moving ahead with general microbiology and immunology. Microbiology, immunology. Right. මේ අපේ group එක කලින් group එකක නේ තාම තියෙන්නේ. ඒකද වෙන එකක් තියෙන්නේ. වෙන එකක් තියෙනවා. මම එක් කරන්න යන්නේ ඒක. හරිද? Paper නෑ තමයි. I click down presentation. So anyway, so general microbiology and immunology. So what exactly this is? So microbiology. So this is in microscope, right? That is something that we can't see, right? So that's what we are talking about. So we can't see that, and then so then how do we respond as a human body? How do we respond? That is like our tolerance capacity. So that's what we are talking about under immunology. So this is a long series. So today we are starting that. Well, basically, at the first place, we need to know what microbiology is. Well, when it comes to organisms, we have macroscopic organisms and microscopic organisms. Macroscopic කියන්නේ අපි වගේ තන්දාල බලපුවම පේනවා. Microscopic invisible to human eye. ඒ නිසා අපි ඒක බලන්නේ මොන හරි enhance කරපු එකකින් basically by using microscopes. So basically when it comes to microbiology, we are studying about the microscopic organisms. That means we can't see with the naked eye. We study them, we look at them by using microscopes. So those are microscopic organisms, basically microorganisms. Why can't we see them? Yeah, too tiny. We can't see them, too tiny, right? Then we can't see them with the naked eye. We study them, we see them, we study them, we analyze them by using microscopic observations, right? Well, where do we use this microbiology? That means microorganisms. Are they useful? Basically, are they beneficial and are they harmful? Yes, both aspects are there. Well, anyway, they're using this microbiology, this study in medicine, right? In medicine, obviously, right? We are getting infected, right? Most of the cases we are getting infected, certain diseases. Then we need to control them. A lot of things. So in that case, we use microbiology in medicine and also in agriculture. So medicinal applications we already know in medicines, like definitely medicine. Entirely we know about we deal with microbes, no? Viruses, bacteria, fungi. We are familiar with this. In agriculture, what are the uses of, how do we use microbiology in agriculture? What are the things that you know? What do you remember about microbes when it comes to agricultural areas? are we really using, and what are the areas? How do we deal with microbes when it comes to agriculture? Any example that you can remember? Agricultural vegetables, honey, මොනාවගේ හරි use කරන තැනක් මගද? Microbiology, microbes, bacteria, fungi, viruses use කරන තැනක්. When it comes to food and beverages, මතක example එකක්. Yeast, yeast, yeah. මොනාවද use කරන්නේ ඒක? Example එක කිව්වා. application එක කියන්න කවුරුද? Fermentation, මොනවා fermentation? yeast කාල ferment කරන මොනවාද මතක් වෙන්නේ? ගොඩක් ඒවා තියෙනවා. yeast කාල ferment කරන මොනවාද? bakery industry, ඊට පස්සේ කසිප්පු industry, ඒකට yeast වදාන්නේ. අර sugar අර පල් වෙච්ච පළතුරු කොහොම ටික දාලා yeast තමයි දාන්නේ. ඊට පස්සේ 1 week time තියලා කැලේ පෙරලා අරගෙන ඊට පස්සේ distill කරනවා නිකන් production එක ඉවරයි. label එකක් නැහැ. ඒක විතරයි නේද? Right. Anyway, good. Yeast fermentation. තව කොහොමද example එකක් ගත්තොත් mushroom. අපි මොකක් කරනවනේ? Right, mushroom. That is fungi, microbes. So in agriculture, හැබැයි agriculture එකේ පාන්නේ bakery industry නේ. හරි example එක agriculture එකේ use එක හරි agriculture නොවුනට that's somewhere we are using microbes. ඊට පස්සේ mushroom ඒක agriculture තමයි. ඊට පස්සේ කසිප්පුත් agriculture නෙමෙයි. අයි example එකක් තරදී. අපි දෙන්නම කියපු දෙකම right. ඊට පස්සේ what else? Hybrid plants. දැන් අපි ගත්තා පස්සේ bee එහෙම අර දැන් අර මාස 3න් හොයන් කපන්න පුළුවන් bee තියෙනවා නේද? ඊට පස්සේ ඉස්සර මාස 6ක් විතර යනවා. And also when it comes to certain fruits අර දැන් අඹ එහෙම ඉක්මනට අර අවුරුද්දෙන් ගෙඩි හැදෙන ඒ hybrid versions. ඒතකොට ඒවට genetically modify කරන්න bacteria, and so we are using bacteria to modify the genes, right? So in those areas, so for making genetically modified plants, right? So we are using microbiology. Industries, ඒකට අපි කියපුවා හරි. bakery industry, කසිප්පු industry, for the industries. And also environmental science. Environmental sciences in the sense like, think about a country where we have a lot of industries. Industrially, now Sri Lanka doesn't have that much of industries. Think about a country with a lot of industries. And that's the very reason that Sri Lanka is still in this economical state. Like, so we haven't gone to industrial economy yet. I mean, production economy yet. Up to that much. What do we have? Tea, rubber, coconut. අපි පොඩි කාලේ ඉස්කෝලේ යනකොට මොනවද ඉගෙන ගත්තේ? තියෙන industry මේ tea rubber pole. තාමත් ඕම තමයි තියෙන්නේ. ඒතකොට we have to go beyond that. Industrially we have to go beyond that. Otherwise we can't be having a grown economy. So that is that. Well, so industries, when we have industries, right? Industrially, what are the problems? Whenever we do industry, මතක දන්නේ Coca-Cola factory එකේ issue එකක් තිබුණා around 2 years before. කැලණි ගඟට තමයි ඒගොල්ලන්ගේ වතුර යවන්නේ. මතකද ඒ issue එක? මතකයි නේ? හරි. ඒතකොට දැන් Coca-Cola කියන්නේ දැන් ඒක අපි ගත්තට පස්සේ අපි දන්නවා windscreen හොදන්න පුළුවන්. bathroom හොදන්න පුළුවන්. ඒ ඉස්සරහට chemicals ගොඩක් තියෙන drink එකක් වයින්. ඒතකොට එහෙම එකක් කැලණි ගඟට යනවා effluents ඔක්කොම ටික. ඊට පස්සේ කැලණි ගඟෙන් තමයි entire කොළඹ වලට වතුර දෙන්නේ. කේස් නේ. ඉතින් ඕක ලොකු ඉෂුව එකක්. ඒකෝට ඔන්න ඔය වගේ. That is an issue. What is the issue? When we have the factory, when we have the industry, right? Whoever the owner of the company, like if the industry is the factory is situated in Sri Lanka, we have the problem. What is problem? The product. Whenever it comes to an industry, we have the product and we have byproducts. Right? Industry in Coca-Cola, byproducts in, right? What industry? We have products and byproducts both. Then product is collected, byproducts should be thrown away. But it is not easy to throw away the byproducts. Right? We have to find ways and means. If it is gas, so gas has to be emitted. We have to analyze what are the gases. We have first analyze what are the gases. In gases, if those gases are harmful, we have to convert those gases into either into less harmful gases or totally harmless gases, if possible. If it is a solution, we have to deposit certain harmful ions and then send it to, right? Harmful ions, when I say harmful ions, I remember one plant that is kangkung. Kangkung, කොහෙද හැදෙන්නේ? ඉතින් හැබැයි මේ ඊට පස්සේ මිදුලනේ නෑ ඒක නෙමෙයි. ඉතරක් හැදෙනවා. කංකුම් හැදෙන්නේ බලන්න දැන් ඇවිදන් යනකොට මේ කුණු ඇලවල් තියෙන්නේ. හරි අපිට තලක් තියෙන්නේ. හරි. ඒ ඇලවල් ගාව බලන්න නෝටිස් කරලා කංකුම් පොලේ සයිස් එක මොකද්ද කියලා. අන්ටි පොළ තියෙන්නේ. අර අපි කැමරට ගන්න පොළ අර පොලින් ගන්න එක වගේ නෙමෙයි. අන්ටි පොළ තියෙන. සමහරවිට අර වට්ටක් පොළ විතර ලොකු පොළ. පරිතියාගට ඒ කංකුම් කියන්නේ the environmental pollution measured in some kind of a unit. If we have like very highly grown kangkung plant, that means there are so many harmful ions just like lead. Lead is carcinogenic, you already know that, cancer causing. Lead 2 plus, mercury 2 plus. Likewise, there are harmful ions. Think about a factory which releases those harmful ions to a water reservoir. Then water contains high amount of mercury 2 plus, lead 2 plus, again. Think what happens. Then what happens is these plants which are ready to consume these ions, they will grow. Then kangkung plant it grows like very high. High extent they could have, right? So then that is an indication that area contains, whenever you see a big kangkung plant, remember there is an indication to see that this area contains high amount of heavy metals, heavy metallic ions, right? So then those concerns are there. So those are the problems I was talking about, the issues, how to handle these byproducts issues when it comes to the chemical industry. When it comes to chemical industries, actually, Sri Lanka had sulfuric acid industry sometime before. The area called Parantan, that's north. The LTG areas, those areas. So then they blasted the factory. Remember, like sulfuric acid industry, it is the most profitable chemical industry in the world. Now, graphene industry is there. You know what graphene is. So sulfuric acid is going to be a very profitable industry, very highly profitable industry in the world. When it comes to chemical industries. So Sri Lanka, we had that in Parantan area, that's like Palali, that side, Parantan area. So we had, Sri Lanka had that factory and LTG blasted that factory. So after that, we don't have that factory anymore yet. So we'll see whether the new government will start the factory again. It's good if they start because the profit is really high. But the capital for the industry is also high. That also we have to admit that. For starting chemical industries, we have to have a big capital. Major use of the big capital is to handle the byproducts. Byproduct is the one that I was talking about right here. When it comes to industries, it's a big challenge. It's a very big challenge how to handle these byproducts. In those cases, we are using microbes to decompose these ions. There are microbes which use these heavy metal ions as like some food source. In such bacteria we can grow, then we can send those factory waste water into a tank. Inside the tank, we can grow that bacteria, and then bacteria will consume those heavy metal ions. Very right. So that is how it is going to be. Right, industries. So that I talked about under environmental science. So in environmental science, we are using microbes in modern chemistry, right? Well, understanding microbes helps us to improve our health, protect the environment, and so basically, for us to have an idea about how healthy we are and how to be protected, that also, like we have to have an idea about microbes. Let's say there is some food open, then we need to know, right, okay, open, so the microbes can easily come, right? Doesn't mean that if we close it, microbes never come. No, we can minimize it, right? We can minimize it before we put it into the fridge. Right. So those are the areas where we are using microbes and microbiology. Right. When it comes to these microbes, that means the organisms which we can't see with the human eye. Bacteria, viruses, fungi, protozoa, algae, archaea. So not only these, there's a lot. I just took some examples only so that you can have an idea about what sort of a thing this is, right? Well, out of these, what are the familiar ones? Bacteria are familiar with me. Virus done on fungi done, protozoa not that much. Algae done on it. අර බීර වැවේ එහෙම අර දැන් අර පොළ පාට තියෙන බීර මතක් වෙන්නේ අර දැන් ඊයේ පොළකට අප් සෙට් එක ඕපන් ඉතින් මේක සෙට් එකක් ආවේ නේ 40ක් විතර ආවේ ඉතින් ඉන්න සෙට් එකක් ඉන්නම් කියලා ටීවී එකට වෙනනම් හිටියේ 40ක් විතර right ඒ සෙට් එක ඔබට හිටපු එකේ ඒ තියෙන අර පොළ පාට තියෙන්නේ ඇල්ගි හරිද so that is that yeah, I was so happy to watch that news. I was waiting to see, like, okay, what sort of response will come. Organizers, right? Everything and talk with the right. Correct. So those are the types of microbes. Well, when it comes to the history of microbiology, right? It wasn't easy. It wasn't easy to have an initiative for microbiological experiments because this is something that we can't see. Anton van Leeuwenhoek. So that was one of the scientists where you see, look at the history, like we have been humans, we have been dealing with microbiology for a very long time, right? 1670s, right? Centuries back. Right, we were dealing with that. So, so then by that time, they start, observing and dealing with microbes, right? It was started by that time because, when it comes to milk, curd, yogurt, that subject, we need microbes, right? Okay. Then after that, so then anyway, Anton van Leeuwenhoek, like, so he started observing and describing, microbes and microorganisms by using simple microscope. Remember, even by using simple microscope, we can't see just one fungi, one cell. It's difficult to see. We see colonies. I think we talked about colonies a bit under chemistry up to some extent, right? So, like that. Then Louis Pasteur, next scientist. There was something called spontaneous generation, right? Spontaneous generation. Spontaneous generation. Okay. Ibe හරිද? දැන් concept was දැන් මොකක් හරි අපි දැන් හිතන්නකෝ මොකක් හරි කෑමක් තිබ්බ කියලා කනුව ඉබේ හැදෙනවා. right? ඔක කනුව හැදෙන්නේ ඉබේ කියලා තමයි concept එක තිබ්බේ. So then experiments were going on for a very long time. Finally, Louis Pasteur, he managed to prove that there is nothing called spontaneous generation. Right? It is happening with a microbial process. That's how it was proven that there is nothing called spontaneous generation. That is, nothing is coming from the sky. Right? Okay. Then Robert Koch, another scientist, then he approached, he approached with microbes to certain like disease level approach, right? So how to deal with diseases and microorganisms, and so then he came up to like certain level, some medical approach, right? So that's how in the history this happened, right? Well, this microbiology, it's a very wide area. It's a very wide area. So that when something is so huge, always what we do is we classify, we categorize the particular area, particular subject into several, so that we can study about it easily, right? So when we study about bacteria, we refer the terminology is we refer that as bacteriology, bacteriology, and virology, that is study about viruses. Mycology, fungi. Parasitology, when we study about parasites, right? Phycology, it's about algae, right? So under immunology, we are talking about, we study about immune system and responses. Basically, that means when we are under attack, when we are under attack of microbes, then we talk about like how our responses of our human body. That is about immunology. Well, microbial genetics. Well, that is about study of genetic makeup microorganisms. It's some very modern technique. We modify the genetic codes of microbes with certain purposes. බැලුවද මේ රීපර් බැලුවද ජුරාසික් පාර්ක් වල අලුතෙන් නෑ ඒකේ T-Rex වෙනකොට ඉන්නේ T-Rex නම් හෝල් එකේ තියෙනවා මම දැන් මීටර්ස් එක දුන්නා මේ අන්න genetically modified dinosaurs, genetically modified ඒ මූවි එක T-Rex එක තරම් ගති නෑ boring හැබැයි ඉතින් right, it's good. So anyway, so genetically modified. So that is microbial genetics. Genetically modified animals, right? Okay. Industrial microbiology. That is definitely about when it comes to industries. Like I was talking about before also, about how to manage the waste products, the garbage side, that we analyze and do with the help of industrial microbiology. Medical microbiology, that's definitely really important. It's about human diseases and how to deal with human diseases and total stuff. Okay, no? Right, that's a terminology. You know that? chemistry කොහොමද පොඩ්ඩක් නිදිමතත් එක්ක පොඩි link එකක් තියෙනවා නේද තාම? නෑ කලින් lecture එකක් free නේ ඒකත් lecture තිබුණේ නෑ නේද? rest එකේ ඉඳී. ඒකට ඉන්දියන් excuse නෑ අද. අරක නයිද හරි back to back lectures lecture එක තිබ්බා කෑවා අපෝ මේකට ආවා හරි පොඩ්ඩක් hectic schedule. දැන් අද එහෙම නෑ ඒක free. Right. Well, microorganisms. When it comes to microorganisms, we have advantages and disadvantages both. Which one is more depends on the situation. Sometimes advantages like, okay, that outweigh the disadvantages. Sometimes the disadvantages that outweigh the advantages. So like that. Well, so in what are the like positive sides of that? Well, decomposition. That is when it comes to the garbage handling. Now think about, okay, like, when the garbage truck comes, right? We are supposed to sort it and give, right? Polythene separately, glass separately, kitchen stuff separately, like that. So that is how we do. That is how we do. Right. So in that case, organic matter, right? Regarding decomposing organic matter. So those stuff are being decomposed by, microbes. That is how like we at home also we can make compost, right? Fertilizer. And the other thing is nitrogen fixation. What is nitrogen? A fertilizer. But nitrogen is a gas. Nitrogen is a gas which is present in the atmosphere. Something which is present in the atmosphere cannot be absorbed by roots of plants, right? From the atmosphere, the plant leaves are absorbing carbon dioxide. Okay. But that is done by plant leaves with the purpose of doing photosynthesis. In that case, the carbon dioxide is not a fertilizer. Fertilizers must be absorbed by the roots of the plant, not by the leaves. From the roots only, it is going up and going to the certain places. So in that case, nitrogen is something which cannot be absorbed like that in the atmosphere. Nitrogen is present in the form of N2. N2 is a gas which cannot be absorbed by the roots. Then what is happening? This should be converted into a soluble version. Is soluble? Roots වලින් absorb කරන්න පුළුවන්. Gas absorb කරන්න බෑ. And solid absorb කරන්න බෑ. Soluble version එකක් විතරයි absorb කරන්න පුළුවන් roots වලින්. නේද? So this should be converted into soluble version, which is nitrate ion. Nitrate ion. Right. After converting N2 gas into nitrate ion in the soil, then the roots are capable of absorbing that. Roots will be absorbing nitrate ion, not nitrogen gas. Okay. Right. Nitrate ion will be absorbed. Well, since the nitrate ions are absorbed like that, okay, so how it is happening? The atmosphere contains N2. Well, microbes, example, rhizobium, they are capable of converting N2 into NO3 minus, Right. This is what we call nitrogen fixation. Right? Why do we call it nitrogen fixation? Because when N2 is in the atmosphere, it is not fixed. It is like unavailable. It is something somewhere like unavailable. Because roots are not able to absorb it. But when it is converted into NO3 minus, now it is usable, right? Now it is usable. That is what we call nitrogen fixation. And also, bread, yogurt, cheese, the bakery industry that we're talking about. So for those also, we are using for food production, right? Microbes are being used. Biotechnology, biotechnology, production of insulin, antibiotics, right? Antibiotics, we had a separate topic about antibiotics under biochemistry, right? So for that, we are using microorganisms and waste treatment and bio remediation. So in that is really, really important. That is when it comes to the industries, right? So these are the positive sides, basically, in simple words, the advantages of microorganisms. Well, we have problems also, not only advantages. When it comes to problems, pathogens, huge problem. They cause diseases to humans, animals, plants, right? Not only humans and animals, plants also, right? Plants, like in agricultural areas, right? We have to take care of plants. We have to take care of vegetables from pathogens by using certain chemicals, right? Mm. Okay. And food is spoiled, right? That's why we need the fridge now, and the food is going to be spoiled. Biofouling, growth of microbes on surfaces, right? Pipelines and all, right? So microbes are being grown likewise. So these are the disadvantages, like in general. There are disadvantages like that. Well, since we can't see microbes, we have to use certain tools to deal with microorganisms. Right. Obviously, we need microscope because with the naked eye, we can't see the microbes. Obviously, we need something for us to see the microbes, which is microscope. Right. Even with microscope, remember, when the microbes are isolated, still we can't see. Still we can't see when the microbes are isolated. In that case, we have to wait until the microbes grow and make colonies. Culture media. What is culture media? Culture media means we supply a medium with all the nutrients that the microbes need. Right? We supply all the nutrition. Again, then microbes are order karana camera medium. Right. Then they utilize all the food and beverages and grow very fast. Right. That is what we call culture media. When they are grown, then we can see. That is culture media. Sometimes, even if they are grown, still we can't see. Still we can't see. Then we have to color them. We have to color them. Right. So those are like we need staining techniques. I think somewhere we made gram positive, gram negative. I can't remember what topic was, but we meant that somewhere, right? So that is for us to see staining techniques and also sterilization methods. That's really important. Sterilization methods. Why? Because microbes. When we are dealing with microbes, definitely we have a disadvantage and we have a risk of getting exposed to microbes because microbes are like we are getting infected. That problem is since we are getting infected, we have a problem of being sick. To avoid that problem, we need sterilization methods, right? Uh, the most familiar now we are used to sterilization methods. With COVID-19, we had that issue, right? Before COVID-19, did we use sanitizers? No, no, right? But after that, uh, it became a situation like now we are familiar with using sanitizer. Like we have this small bottle in the car, always like still in the bag, go somewhere, right? So then, for sterilization methods are being used. Mm, right, right. Well, sterilization, that is about elimination of all forms of life, including spores, that means, you know, when it comes to microbes, the grown bacteria, fungi, virus, or whatever, like the grown version is there. And spores, right? Just like eggs. So then the particular bacteria, fungi, virus is not there, but the spores are there, like egg-like, the initial growing unit is there. Still not a fungi, right? So that is also there. So elimination of all of them, right? Full package, full package. That is sterilization. Then our whatever the liquid should be powerful enough, right? Right. And aseptic netting, practice to prevent contamination during experiments or procedures. Uh, what do you mean by that? Contamination. Now, as an example, let's say you're going for a blood test, right? The needle, the syringe that which is being used for taking blood from you is definitely discarded. Can't use it again, right? For another patient, so contaminations will be there, right? Blood sample, urine sample, a serum sample, whatever. Right. We have to definitely prevent the contaminations. Otherwise, wrong results are coming. So, aseptic techniques. Those are aseptic techniques. We should be careful about those also. Hmm. Right. Sterilization. Sterilization now, think about a surgery or something. That is, in a surgery, it is not about now taking a blood sample or something. Yeah, it is about aseptic technique should be there. In a surgery, sterilization has to be there. Uh, we have to sterilize the equipments, the scissors and everything, knife, right? You have seen in movies also, like, think about in a jungle or in a ship or somewhere, like some surgery has to be done. Uh, so minor scale surgery, you have seen in movies also, like, they are using some like whiskey or vodka or something, like some liquor to sterilize uh, those needles and stuff, right? Because alcohol also is a very good sterilizer, right? Uh, since alcohol is a very good sterilizer, only in COVID season, I wonder whether you remember in some country, like people went to illegal liquor place and drank. 16 people died on the spot because that was toxic. That alcohol was toxic. Mm. Right. Well, when it comes to human health, well, beneficial microbes in human body. So when it comes to microbes, right? What is the idea that we get in human body? Don't we have microbes? We do. Microbes are everywhere, right? So even on skin, right? Microbes are there. These microbes, there are interactions between the microbes. Microbes, there are interactions, right? Then, so whenever we break that equilibrium, then we have a problem. When we remove microbes, right? Yes, let's say that okay, because of something that we apply, one of the microbes is getting removed, then it is going to cause a problem because then since that combination is not there, another microbe will grow on the skin. Then that will be a problem. Which is why this is referred as allergic conditions. Any you know, uh, do you have that experience? Like, when you apply some, we'll say face cream, right? Why some face creams are allergic to your skin? So then you refer that skin as sensitive skin, technical term, sensitive skin. අර cream එක කෑවම අවුල්, right? But what is happening is there because of the pH value, or maybe some content which is present in that cream, certain selected microbes are removed from the skin. Now, that particular microbe is not present on the skin because it was removed by the cream. Then the other microbes which were not damaged by, which were not eliminated by that cream, they are growing. They are growing. Okay. Since they are growing, now there are problems on this. Right? Competitive is good. Right. So that is there. Pathogens. Right? Pathogens. Those are the microbes that cause, those are microbes which infect us. Pathogens. Vaccines. Vaccines is a solution for those pathogens. They prevent microbial diseases. They prevent microbial diseases. Antibiotics. They treat bacterial infections. Bacterial, okay? Right. Viral cases, that's a different story. So we'll study about that separately. Right. So when it comes to human health, these are the aspects that we are dealing with. Right. Microbiology, with microbiology, where are we going? Right? What are going to be the future trends? Where are we going with microbiology? Right? Where are we going with microbiology? Maybe I will in there no yeah. So industry wise microbiology එක අපි පහට දෙන්නේ නින්ද යන එකට අමතරව හරි එහෙම ගත්තොත් antibiotic resistance. What do you mean by antibiotic resistance? Antibiotics අපි කොහොමද බොන්නේ? පැන්නෝ කරලා අපි දැන් පැනඩෝල් නම් ඔළුවේ රිදෙන්න ගත්තා පැනඩෝලයක් බොනවා. Antibiotics අපි ඒ සිස්ටම් එකට බොනවාද? පැනඩෝල් බොන ක්‍රමයට නෑ. මොකද පැනඩෝල් ගන්න බොන විදියයි antibiotics බොන විදිය අපි ගමු amoxicillin. tetracycline. තමයි අපි antibiotic බොන ක්‍රමයේ පැනඩෝල් බොන ක්‍රමය අතර වෙනස මොකක්ද? ඒ දෙක අතර වෙනසක් තියෙනවා නැත්නම් එක විදිහම මේ ආරකක් බොන්නේ. මම කියන්නේ මම කියන්නේ බන්නේ අරකට වතුරත් එක්ක බොන්නේ මේකත් වතුරත් ඊට නෙමෙයි මම කියන්නේ. වතුරත් එක්ක බොන එකට අමතරව ගත්තා පස්සේ frequently 5 days හරියට antibiotic is a course, right? 3 days course, 5 days course. Panadol බොන සිස්ටම් එකට නෙමෙයි බොන්නේ. ඇයි? Any idea? We are under the point antibiotic resistance. Yeah. That's right. Now, the particular microbe by which we are infected, right? We are taking the antibiotic, right? Now, let's say, okay, sometimes we are, smarter than the doctor, no? So in such cases, what do we do? දැන් හොඳයිනේ. දවස් 3කට බෙහෙත් දුන්නා එක දවසකින් හොඳයි. right. දැන් දවස් 3 දවස් බොන්නේ. right? හැබැයි හොඳයි නෙමෙයි. By the antibiotic, the particular situation is controlled. Right? Controlled. Right. Now what is happening? We stop taking the second and third day doses because we feel like now we are fine. No, we are not fine. What is happening is now whatever the pathogen, whatever the bacteria, fungi, whatever the microbe, what is happening? Whatever bacteria, so that these microbes are getting modified. That is getting modified. What do you mean by modified? Now bacteria knows that okay, this is a medicine which is being given, right? Before we kill the entire thing, we stop medicine. Now bacteria is getting adapted to that. Let's say amoxicillin. Amoxicillin අපි ගත්ත දවස් තුන නේද? එක දවසින් නැවතුවා, right? Now bacteria knows about, right? Chemically, bacteria knows about amoxicillin. Now, let's say bacteria wasn't killed completely. Now, bacteria, which is inside us, is modified, adapted, and resists. And it resists amoxicillin. Right. Now, what is happening? Next, okay, now again we fall sick after a few days. Now, okay, one day we feel like recovered, and second day, third day, we did not take the antibiotic, right? So then it takes now this bacteria. Now bacteria is under control for some time. Now it wasn't completely killed. Then after like another 3 days, okay, now we are infected. Not that we are reinfected, we were already infected. Now the bacteria, the same bacteria that was inside the body, is modified and working. Now again we take the same. We go to the doctor and again amoxicillin is given because the doctor starts with the like, okay, with the situation, okay, amoxicillin is given. It doesn't work. Why? Now the bacteria, whatever the pathogen, resist amoxicillin. Then, of course, but we get there, right, okay, those doctor gave this medicine, okay, it didn't work. We come to a conclusion, right? That is a conclusion that we come to know. The doctor was left with no choice. And he is supposed to, medically, he is supposed to start, like ethically, doctor is supposed to start with the lower strength. But there are some doctors, they don't start because they know that situation. So marketing wise, they feel gathering some reputation in the society that he is so good. The one shot කියලා doctors ලා. එහෙම හැමදාම දෙයකම ඉක්කින්නවා. හරි one choice. So they give something which works for every single thing. That's a very strong dose. Then people think that okay, whatever you go to one shot, problem solved. Because I behave hurry. Right? Right? Because that fellow is smart. He knows that okay, the people do like this, and he knows science, he knows chemistry, and he knows that okay, right, okay, we shouldn't start from the ethical background, medicine, that's all chemicals, right? So ethically, it is not right to give the strongest possible thing to a patient. You never do that. But there is something like, in the society, now there's nothing called ethics, no, right? People do whatever they want to do. Just to forget about medicine, just look at the road, how they drive in the road. And so that is a situation like so. Ethics is something which is not present, no, right? So then there are doctors, they prescribe the highly possible, the most strongest one. So anyway, so what do you mean by antibiotic resistance? If we stop the antibiotic in the middle of that journey, it resists. It resists the action. Right. Then that particular antibiotic doesn't work. Right. That's why, in some countries, when you get sick, they don't give medicine. Now, in Sri Lanka, to some extent, we can buy medicine, antibiotics over the counter. Right. Officially, there should be doctor's prescription for us to buy medicines. But are we really doing that? And right? So then over the counter also we can buy. But there are countries like you can't buy medicine like that. Even if you go to the doctor, they give the like mild, the least possible one, mild one, right? As an example, Australia, right? Until like unless you are really sick, you don't get like proper medicine like in Sri Lanka. So that is to prevent like globally that problem is enough. Let's say that as an example, let's say you get sick and uh, you stopped your prescription in the middle of that journey, not 3 days, not 5 days, like one day you stop. And so then now you are getting sick again. And that virus is spreading to him, we'll say. Now, for him, what he is getting is not the original version that you got. Neither he is getting the modified version. Then can he start with amoxicillin? No. Amoxicillin doesn't work for you also. And for him, he wasn't sick before. But for now, like right now, amoxicillin doesn't work for him because he got a modified version, which is resistant to amoxicillin. Then he has to start with maybe augmentin, cyclosporine, something like that. So that is like, it is harmful to you and it is harmful to rest of the society also. Then, we have to, when we take medicine for certain diseases, like we have to have certain knowledge about that. Either we should follow instructions properly, or else we should know why are we getting that instruction. So the antibiotics, Panadol one system, right? Because there is something called antibiotic resistance. Okay. Then microbiome research. So this, when it comes to this antibiotic resistance, then we are doing research to find out what modifications are being done. Right. To this particular modification, what should be the particular solution? What antibiotic should be given? I think the best example to take is COVID-19. In that case, remember the original at the very beginning, COVID was one thing. It was say alpha beta beta. Something I can't remember. And that's why, like, remember, we had to go for 3 vaccines. First one, second one, third one. Take another China and minister got caught also like for like fake bills and stuff. Right. So, the research should be done analyzing the modifications of the bacteria or fungi, whatever the microbe, it should be properly analyzed, right? It should be properly analyzed and should be taken care of. Okay. Then, after the research, then synthetic biology. What is that? Okay. Bacteria, version number 1, it got modified to version number 2. Then the research was done. And after the research for, the modification version number 2, then we have to synthesize a particular antibiotic according to that situation, right? That is synthetic biology, right? Bioterrorism prevention. What is bioterrorism? Any idea? Bioterrorism. Terrorism we know, right? Terrorism we know. What is bioterrorism? Do you remember when it comes to, if I go back to the same example, COVID, you remember there was a very big argument that this was created in China. China was release right? It wasn't proven that it was produced in China, but there was a like big argument that China will make a mother, and countries other than. It was like that point was supported by China, made the antibiotic also. No? The vaccine was made by China, Sinopharm or something. Right. So, bioterrorism is that. I'm not saying that China did that. So then in World War II, this was being practiced, right? So then Vietnam was diseased, right? And, and yeah, so then Vietnam, like US planes were going over Vietnam, and so they were releasing these bacteria, modified bacteria and stuff, right? So that the people get sick, they can capture the land easily, right? This is bioterrorism, right? Then to prevent bioterrorism, also we need microbiology. Bioterrorism is created by the sea microbes, and so the solution also should be created by using microbes. It's just like missile and the anti-missile missile guard, like that. Pandemic situations, yeah, COVID-19, yeah, that's what we're talking about. So this is where we are heading with microbiology. Right. Well, now let's come to, now we have a clear idea about microbiology and microorganisms. So when it comes to types of microorganisms, well, microorganisms, the organisms which we can't see with naked eye, right? They exist as unicellular, unicellular. That means one cell, and multicellular, right? Multicellular. That means more than one cells, and also cell clusters together. They make colonies like these clusters, right? So in those ways, microorganisms exist. Well, we'll come to bacteria. Well, when it comes to microorganisms, we are using two terms. Prokaryotic cells and eukaryotic cells, right? Prokaryotic bacteria, they are coming under the category of prokaryotic cells. What is that? No nucleus, no membrane-bound organelles, right? There's no nucleus. Then where do we have DNA? Their DNA is in cytoplasm, not in nucleus, right? Which is why we are using bacteria for genetically modified experiments, because in the cytoplasm itself we have genetic material. Hmm? Right. Okay. So those bacteria, it is coming under the category of prokaryotic cells. But we find bacteria in soil, in water, and in living organisms, right? Okay. When it comes to shapes, when it comes to shapes, shapes, of course, we use certain Latin terms about shapes. Foci, that means spherical, round ones. Right. Bacilli, that rod shape. Spirilla, that's spiral ones. Those are the shapes of bacteria. And also, when it comes to bacteria also, that is common, we have the common feature. Either this could be beneficial or harmful. That is, both the ways are possible, right? Can be pathogenic. Pathogenic means it can be harmful. Or also, it could be beneficial also. As we discussed before also, nitrogen fixation possible and fermentation possible. I was talking about the shapes. When it comes to the cocci, that is going to be the shape is a round shape, spherical, and you must be wondering, okay, round shape, spherical, is this spherical? No, that is not spherical. But remember, it is a combination of lot of spheres, right? Football, like right. And this is also a combination of balls. And here also, yes, balls, spherical. Those are spherical ones. And bacilla, that's rod shape. Now, that's not a sphere, you see, rod. Now, this is a single rod, right? And here, collection of rods. Collection of rods. Spiral shapes, right? This is the spiral shape. So that's totally different from the rod and spheres, like this. Fungi. Fungi, that's not prokaryotic. This is eukaryotic, right? So examples, yeast, moss, mushroom, right? And it has a cell wall, a thick one. Cell wall is made up of chitin, right? Cell wall is made up of chitin. Well, fungi, we can use them as decomposers, but remember, decomposers is good, but that's an advantage. But some are causing diseases as well. You might remember uh, from pharmacy, certain cases we buy antifungal cream. Antifungal cream. That is to prevent the fungal infections. Fungal infections are coming to the skin most of the cases, right? Rashes වගේ දේවල්. So we apply antifungal cream, right? Hydrocortisone, candy, already, right? Those are antifungal creams. So yeah, the shapes. We have different shapes. Looks nice, right? Right. This is just to compare the difference between, appearance-wise, the difference between bacteria and fungi. This is bacteria, and this is fungi. Yeah. What's the major difference that you see between bacteria and fungi? Appearance-wise, bacteria, it's like, um, like in the cellular level, like, uh, it's not that aggregated, right? It's not that complex. But when it comes to fungi, over there, you see, it's a collection of load of cells, and aggregate together to produce a big structure. Yeah. Right. So structure-wise, look-wise, basically appearance-wise, well, this is how we see the difference between bacteria and fungi. Another category is protozoa. Protozoa. Maybe protozoa is not that familiar. Maybe you have heard of, right? That is also coming under the category of eukaryotics. That is unicellular, only one cell. Right. They are mostly motile. That means they are movable. Right? Movable. Right. For them to move, as an example, let's say now for a fish to move, fins are there, right? So then for protozoa to move, right? They don't have fins. They're not that organized. They don't have fins. They have like fin-like objects, flagella, cilia, pseudopodia. Pseudopodia means this is Latin. Pseudopodia, just like a leg-like structure, a leg-like structure. Ah, thank you. right? It is a leg-like structure, pseudopodia. Mm. So basically, that's another point. They have something for them to move. They have something for them to move, right? So they are moving. They are moving. That means they are food and beverages, whatever the food requirements and the nourishments they are taking from the environment, where they are moving, right? Structures of protozoa. Do you see this? Like so, then yeah, these are the ones they are using for them to move. අර දැන් අර ඔරුවක අර හබල් ගාලා යන රෝබින් ඒ සිස්ටම් එකට තමයි මේ යන්නේ. right? දැන් සියුඩෝපෝඩියා මේ තින්නේ කකුල් වගේ ඒවා. කකුල් නෙමෙයි. හරිද? ඒ වගේ ඒවා. ඒ ගැජට් මූව් කරලා මෙහෙම යන්න පුළුවන්. flagella. right? For them also we have. Right. Algae. It is another category. We have photosynthetic eukaryotes. That means like so they actually they don't have to move because they're photosynthetic. When sunlight comes, they can photosynthesize and produce food. So then there is no requirement for them to move. Then what is their requirement? There has to be sunlight coming. If sunlight doesn't come, right, they can't move. Right. So now they are moving. And also, they produce oxygen, right? Because photosynthesis is being done. Oxygen is released. That's about algae. Right. This is diagram. Like what is in green color, what is being floats here. All algae, right? Right. And therefore, they do photosynthesis. Viruses. Obviously, they don't have a cellular structure. If something is a cell that works like survive independently, that survives independently. But virus definitely needs a host cell, right? They need a host cell. Just like some politicians, they need some leader to jump into that political party and talk a lot, right? Without a leader, like so they just stay, right? So then so they are proper viruses also, right? Right. Now in this case, you see, viruses also like that. They need a host. Without host, no, that virus is no one, right? Which is why we say they are acellular. They don't have a cell, right? They are acellular. Cellular means, yeah, it is a cell. Acellular, it doesn't have a cell. Right? Well, so it has, but though it doesn't have a cell, it has DNA or RNA enclosed in the protein capsule. Then what happens when that enters? When that enters to a host cell, maybe human cell, maybe animal, maybe plant, what is happening? In that case, that will be growing and multiplying. Right. Virus structures, you're very highly familiar, right? COVID-19, it is this shape. This is a virus. Well, when it comes to pathogens, not only that, pathogens are harmful, no. Beneficial or harmful, either beneficial or harmful, it is really important for us to know their nutritional level. I mean, what do they eat? Their nutritional level, their enzymatic activity, their metabolism, their growth. We need to know. Why? If it is a beneficial one, it can decompose or useful level. Now we grow correct. In that case, we have to supply the relevant nutritions. In that case, we have to supply the particular chemical background for their enzymatic activities. What background? We know enzymes are pH sensitive, temperature sensitive. So all those like favorable conditions for enzymatic activities, like we learned in biochemistry, we learned about metabolism, anabolic and catabolic pathways. Then we have to support them. Enzymatic activities, metabolism and growth. And that is in the beneficial side. Think about this, it's a pathogen, it's a disease. Okay, so in that case also, we need to know their nutritional level and everything, nutritional level, enzymatic activity, metabolism and growth, so that we can eliminate them, right? So let's say if we study the conditions, favorable conditions for their enzymes, then if you remove those conditions, yeah, then that particular pathogen can't survive it. And growth. Mhm. Well, when it comes to microbial nutrition, we need to know certain factors. Well, just like for us, for them also, there are macronutrients. Macronutrients means they need those. They need those in large extents. Now, as an example, our food, we eat rice a lot, and meat, vitamins like so up to some extent. Right, so we don't everything in equal proportion, isn't it so? Right? For them also, macronutrients, the elements, the nutrients that they need in large excess, large quantities: carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, magnesium, calcium. Those elements they need in high quantities. Right. And also the stuff that they need in small amounts: iron, manganese, zinc, copper, right? In small amounts. Well, and in addition to that, there are growth factors as well: vitamins, amino acids, purines, pyrimidines. Those are the growth factors. Well, if we need this particular bacteria to grow, we supply. If it is harmful pathogen, we remove it. That is why, when you are sick, we all have heard like when you are sick, depending on the disease, we know it is not good to eat these stuff. නේද? අර තියෙනවනේ. ලෙඩ වුණාට පස්සේ දැන් එහෙම යෝකට මේවා කන්න හොඳ නැහැ. ඊට පස්සේ මේවා කන එක හොඳයි. Right. So then why is that? Certain food stuff, like certain food stuff, it contains a lot of nutrients which bacteria also needs. So අපි ඒවා වැඩිපුර කෑවොත් bacteria also is growing, right? And more than that, you have heard like some certain food, vegetables and heaty food, සීතල කෑම රස්නේ කෑම here, right? So in certain cases, it is not good to eat these stuff. Certain cases, it is not good to eat these stuff like that. So in such cases, we are controlling certain conditions, certain essential conditions required for bacteria and fungi. Right. Okay. Yeah. So that's about the microbial nutrition side. Well, for bacteria, for microbes, we need to know about for certain, not only for microbes, even for us, there is an energy source and there's a carbon source. Right? There's an energy source and carbon source. Well, for us, humans, okay, energy source, for us, of course, what is our energy source? Sunlight? For humans, forget about this. For humans, what is our energy source and what is our carbon source? For us, for everything, there is an energy source and carbon source. Must be. Because we need energy and we need carbon for a plant, for dogs, for cats, for animals. There is an energy source and carbon source. So you can tell me from where do we get us humans? Okay, not these plants. Us, from where do we get energy? From where do we get carbon? What is our carbon source? What is our energy source? Humans. කොහෙන්ද අපිට කාබන් හම්බ වෙන්නේ? ඉතින් අමාරු ප්‍රශ්නයක් හිතන්න තියෙන ප්‍රශ්නයක් මම අහුවේ. අපි බයෝකෙමිස්ට්‍රි කරා, එක්සෑම් උත් කරා. Carbohydrates, proteins, on the carbon source. We were studying about the elements which are present in those essential food, right? So we get carbon from carbohydrates, from lipids, from proteins. So that's our carbon source. What is our energy source? Sunlight. Louder. Food. That's right, food. Chemical energy. Chemical energy. Right? එතකොට එහා පැත්තේ පාරගණිත තියෙන ගහ. අර කොට්ටක් බගහ. ඒකට carbon source කොහෙන්ද? energy source කොහෙන්ද? carbon source කොහෙන්ද? ගහට. carbon dioxide. Carbon dioxide from the atmosphere. Carbon dioxide. ඒ carbon source එක. ඒ ගහට energy source කොහෙන්ද? Sunlight. Right. So this table is in general about this. About what? About energy source and carbon source. So if the energy source is light and if the carbon source is carbon dioxide, that is photoautotroph. Photoautotroph. Photo in Latin, that means light, okay? So whenever light is the energy source, that is going to be a photo, all right? And when the carbon source is carbon dioxide, autotroph. That is, carbon dioxide is absorbed and it is producing food, autotroph. Right. And if carbon source is some other organic one, heterotroph. Can be heterotroph, right? Hey, අපිට කෑම හදන්න බෑනේ. Right? We buy food and eat. Right? So we can't produce food inside the body. So we come under the category of heterotroph. Right. If the particular species does not depend on sunlight, right? Now we don't depend on sunlight, right? In plants which depends on sunlight, we don't depend on sunlight. Our energy source is chemical, right? So chemical. Then chemo, right? If the carbon source is carbon dioxide, chemo autotroph. If the carbon source is organic, that means some other external food, that is going to be heterotroph. Then that is going to be chemo heterotroph, right? Then we categorize nutritional requirement of anything, not only microbes, by looking at the energy source and the carbon source. By looking at the energy source and carbon source. Right. The nutritional side is done. With the nutritional side, what is happening with the nutrients are there? It's definitely related to the growth. When it comes to growth, remember, there are two factors. One is growth is definitely something increasing. Maybe it is increasing the number of cells. That is growth. That is growth. And also, it is about increasing the size of each cell. That is also growth. Right? Increasing the number of cells and increasing the size of the cells. That is growth. All right. Right. When it comes to growth, how do we increase the size of the, not the size, number of cells, right? When it comes to bacteria, their number increases, their number becomes doubled. If you have one bacteria that is divided into two, then each divided ones are separate cells. They are again divided into two, right? Okay, then one becomes two, then it becomes both are becoming again two and two, and those ones are again becoming two and two and two each, right? So this type of expanding the number of cells is what we call binary fission. Binary fission. That means it is the size, not the size, I mean the number becomes double, 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 like that. So basically, the cell duplicates DNA. Copies are being made. Copies are being made. Elongates and splits into two daughter cells, like parent cell and daughter cells. Initial starting cell is a parent cell. And what is being made, the two new ones, they're daughter cells. Look at this diagram. That shows how binary fission is happening. First, we have the initial cell, parent cell. Then what is happening? Cytoplasm and the nucleus. Nucleus is going to be duplicated, right? And the two equal ones are made. right? Then it is separated into two new cells. Both are exactly equal. And then only this becomes a colony. Then after that, this becomes doubled again. This is divided into two again. Each one's are again divided two by two by two. Then this becomes a colony. And all are exactly equal to each other. Then we see the colony. So singular cell maybe we can't see, but definitely we can see the colony, entire colony. Right. Let's say we uh, we were talking about culture medium. Remember what is culture medium? Culture medium කියන්නේ මොකක්ද? ඒක නේ. culture medium කියන්නේ මොකක්ද? Microbial growth එකට ඕන කරන ඔක්කොම ටික දාපු කැම බීන ඔක්කොම supply කරපු medium එකක්. හරිද? In a petri dish or some other container, if you put everything which is very highly essential for the microbial growth, that is a culture medium. එහෙම culture medium එකකට microbes දැම්මට පස්සේ මොකද වෙන්නේ? Well, you know, at the very beginning, the microbial growth will be very slow. Very slow. Why? Now the microbes that we put into that medium is not that familiar with that medium. ඉතින් අපි උගේන කොහෙද දැනට දාන්නේ. හරි. අපිට කන්න බෝන જાති කොහොම ටික එතනක් තියෙනවා. But you know, it will take some processing time for us to be familiar with it. නේද? අපි ඔකilla දානවා. පිටින් අපිට දැන් අපිට free ඔක්කොම food document free කියලා. අපි කියන තැන් ටික හොයාගන්න ඕනේ. Right? It takes some time, processing time, which is what we call adaptation. Right? So, for microbes also, there is an adaptation time. It has to be familiar with the food we have supplied. It has to be familiar with the temperature given. Right? Normally supply the optimum temperature, optimum pH. Those are the exact values, no? Right? There are differences. So, the microbes, they have to adapt to the temperature, to the pH value, and to the other factors. They have to adapt. And the viscosity of the medium. Likewise. Then, at the very beginning, the growth will be slow. That binary fission I was talking about, growth will be slow at the beginning, even though everything is available in the medium. Right. That range, that period of time, we call lag phase. ඔක්කොම જાති තිබුණාට place එක අලුත් ඒකට තාම බැක්ටීරියා set නෑ. දැන් හිමීට තමයි වැඩේ වෙන්නේ. Right. Lag phase. Then, okay, now it comes to a level, now everything is like familiar, right? Now, of course, that unfamiliar situation is not there anymore. It grows really fast. That is called as log phase, exponential phase. We can see the rapid cell division, right? It's multiply. So we can see a very rapid cell division, right? Now, then it comes to stationary phase. Nutrition depletion, waste accumulation. What is that? You know, whatever the food that we supply, there is a lifetime for food, right? Think about a fish bun. So, okay, you buy the fish bun in the morning. You can eat that in the afternoon also, but you can't eat that in the evening. By that time, it is spoiled. There's a time. That's fish bun. Egg bun, will it survive the same period of time? A kibula bun, a bread, for everything, there is a lifestyle. For this food also, but food, the culture medium food, also has a lifestyle that survives some period of time. With some period of time, right? Then what happens? The nutrients depletion, right? Kind of in our words, it is the food is getting spoiled. And waste accumulation. What is waste accumulation? The microbes that we grow, right? And now they are waste. Like we'll say in terms of human, we'll say urine and stuff. Likewise, they are for them also. There are waste products, right? Metabolic products, waste products will be accumulated, right? So that is where we call it stationary phase. Stationary phase. Sorry, stationary phase. Okay, now the next phase is death phase, right? Cell death exceeds reproduction. What is that? Reproduction, of course, is happening. That binary fission is happening. But the death rate, the cells are dying in a faster rate than that. Why is that? Because now the competition for food is going to be severe. Because food is becoming unspoiled food for the bacteria is becoming slowly unavailable. Right? Food becomes unavailable, and the competition is high. Right? In such cases, for their survival, it is difficult for them to survive. So, death rate is higher, reproduction rate is low. Right? Then, so if you are analyzing a microbial growth, which phase out of these four that you are selecting to observe this? Let's say, okay, let's say you analyze a urine sample of a patient. Okay, you collect the urine sample and you take the proper culture medium, right? And you get it grow, right? Then what is happening? So out of these four phases, like after growing, you incubate it after growing. In which phase are you are going to observe it, right? Because for us to analyze the disease, we should know what bacteria this is. We should know what fungi this is going to be, right? Then we have to observe it. Now, out of these four phases, which phase is the best phase for us to observe? What do you think? First, second, third, fourth. Second. He says second. Agree? Hello. Yeah. First, second, third, fourth. We can't use the first one because still we don't have enough microbes grown yet out. Obviously, we can't use the death phase because now we have dead bodies more than the living bodies, right? Just like a war, now the soldiers are dead now. So it has to be second or third. Yeah, we should select the last part of the second division, right? Second one, because there only we see a rapid cell division. Even if it is the first part of the stationary phase is fine, right? So those are the two phases, right? The last moment of the log phase and the first moment of the stationary phase. That intermediate moment is the best moment for us to take the culture medium and observe the grown bacteria. All right. Right. About the factors affecting on the growth. Temperature. You know, for certain microbes, we need different temperatures, not the same temperature. Right? There are microbes which grow 0 to 15 degrees Celsius. Right. There are microbes. They grow from 20 to 45. Right. There are microbes. They grow above 45. Then when we try to control, the microbial growth, let's say you are in some food research or something, you need to preserve food. In that case, you see, so then you have to analyze and see, right, whether we are keeping this in the fridge, right, 0 to 15, 0 to 15, okay, then by keeping the particular food in the fridge, we can't protect it. Why? The particular bacteria is growing between 0 to 15. Then we have to put that in the deep freezer. Then right lower temperature. That's why there are stuff that we put in the fridge, like the normal compartment and the deep freezer. Right. Okay. So that's about the temperature. pH. pH wise also, we have acidophiles. That means the microbes which survive in the acidic pH values. Neutrophiles, they survive in neutral pH values. Alkaline philes, they survive in the basic ones. I mean, bases, alkaline solutions. Right. Then it is important for us to know whether to prevent this from being spoiled, whether we are supplying acidic medium, alkaline medium, or neutral medium. මතකයි සමහර කෑමවලට අර ඇසිඩික් අර මොනවාද මේවා ගොරක දාලා තියෙනවා ඇසිඩික් ඒක ඇසිඩ් pH වල තියෙන ඒවා ටික ප්‍රෝ වෙන්නේ නැහැ like that. In some cases we supply alkaline medium අර මේවා කහ එහෙම දාලා තියෙන්නේ right neutrophils is neutral medium. Well, that's about pH. Oxygen wise, in that case, also there are several categories. Obligate aerobes, obligate anaerobes, facultative anaerobes, and if I go back to the same example, fermentation of sugar, the liquor industry, illegal one I'm talking about. In that case, the sugar solution, and then we put yeast, it has to be closed and key. Because yeast that grows in anaerobic medium, oxygen should not be there. Oxygen should not be there. Microbes, some microbes, they grow in the presence of oxygen. It's a must. Sometimes they work, some bacteria, some microbes, they work completely in the absence of oxygen. Facultative, sometimes regardless of oxygen is there or not, still it works. Right? Then we need to know about controlling and about favoring the microbe. We need to know the oxygen requirement. Right? If it is about growing the microbe, we need to, we should try promoting the growth, right? Or else, if we are trying to control it, we need to destroy that. Okay, right. Water, anosmotic pressure, right. Halophiles, salt loving, xerophiles, dry conditions, microbes, depending on their like favorite environment. Salt loving ones are there. Dry conditions, the bacteria which survive in dry conditions are there. Like that. Right. So this is the background of microbes. Not done, right? So basically, this is where we are heading to through microbiology. Mm, Anything which you did not understand? I think I should ask that question from those who did not sleep, right? Anyway, remember micro is not hard, and it makes more sense also because we are talking about like the things which we are needing in our day-to-day life, right? All understood, right? Right. Okay, then. With that, we'll wind up for today, and we'll meet up next week to continue with this. Thank you. When is the date today? 7, eh? All right. Okay, then. See you next week.
Ada
Good.